First of all, I would like to thank Professor Sinha, the Vice Chancellor, and the Dean, Professor Malkania, and finally, Dr. Tashveen Ashraf, for inviting me to be the guest of honor in this function. Ever since mankind existed as a hunter-gatherer, there were no food crops. People used to go around hunting animals or various kinds of plants and fruits. Man has been preoccupied with the qualities and the availability of food. Even in ancient times, people were interested in the quality of food that you want to eat or which was available to humans. And one such person was Hippocrates. Hippocrates was a Greek philosopher and scientist, or a doctor of whatever medicine existed at that time. Even today, when the medical graduates pass out, they are given the Hippocrates oath. You must have heard about it, that if when passing out your MBBS, you are asked to take the oath which was prescribed by Hippocrates. And that was 2,500 years back, and that is 500 BC. And what he said about food is there. Let food be thy medicine, and medicine be thy food. See, in other words, he was advocating that many of the diseases can be prevented or cured by various kinds of food items or foods available. So we have been interested in my lab. Well, actually, I'm now retired, but I've been appointed as a professor emeritus. So I still have something to do with whatever research I was doing earlier. And one of the things we have been interested is plant polyphenols. These compounds are considered to be antioxidants. But what we have done is shown that these compounds also possess anti-cancer properties. And I'll show you some data to prove my point, or at least to demonstrate that it is certainly a possibility. Now, polyphenols, some of the polyphenols are listed here that are found in various food items, such as onions. You have a polyphenol known as quercetin. Then in grapes, red grapes, you have a polyphenol known as resveratrol, which is found in the red wine and comes from the skin of the red skin of the grapes, and which is, considered to be cardiovascular, disease preventive, as well as anti-cancer properties. Then we have, which is found in green tea, and then we have finally in this slide, we have curcumin, which is found in turmeric, haldi, which we eat every day in our sabzi or any other preparation. So, there is epidemiological evidence to suggest that consumption of polyphenols in fruit, vegetables, and beverages derived from plants, such as the green tea or other concoctions and red wine, they have a reduced effect on the induction of various types of cancers. There is epidemiological evidence without found in, as I said, in grapes, red grapes, and these are structurally correlated compounds. The first one here is known as transtrilbene. This is not a polyphenol because this doesn't have a phenol group, phenolic group. The other one is resveratrol, the structure of which I showed in the first slide, and then there is another structurally similar compound known as bicetanol, which has an additional phenolic group. So, what we showed earlier in our first experiment was that these compounds, if you they have the ability of hydrolyzing or degrading DNA ions, and this is an experiment to show that this is indeed the case. And this here is transtrilbene, which is not a polyphenol, and this doesn't degrade DNA, whereas the other two compounds, that is resveratrol and bicetanol, they are capable of hydrolyzing DNA. This is an experiment done in vitro in a test tube. So, these are some of the observations which we have found in literature, and then we also observations made in our own lab, and which have suggested to us what this mechanism or cancer preventive property of these compounds could be. Finally, the most important point here is that I don't know whether you can see, of particular interest is the observation that several plant polyphenols are capable of inducing cell death in cancer cell lines, whereas normal cells are refractory to the cytotoxics action. So, this is quite important, that only cancer cells in cell culture are killed by these compounds, and normal cells are not affected. So, using these observations and others in the literature, we proposed a hypothesis. This is the mechanism how this is brought about, the generation of oxygen radicals, but I won't go into the details of that. So this is the hypothesis which we proposed many years back. About 20 years back, we proposed this hypothesis, which says that mobilization of endogenous copper ions by plant polyphenols and consequent oxidative degradation of cellular DNA would be an important mechanism of their anti-cancer properties. Now, remember that I mentioned that traditionally these compounds are considered to be antioxidants. What we are showing, or what we are proposing, we intend to demonstrate that shortly, is that it is not the antioxidant action, but it's the pro-oxidant action, which means it generates oxygen radicals which react with the macromolecules in the cell, particularly the the DNA, cellular DNA, leading to cell death. So that's the hypothesis. So this is another series of polyphenols found in green tea. Now green tea is drunk quite extensively. In fact, almost everybody drinks green tea in countries like China and Japan, and it is now also becoming quite popular in our own country. And the difference between green tea and black tea is that the green tea leaves are fermented, and the fermentation process leads to chemical changes and polymerization of many of the polyphenols found in the green tea, which gives the particular color and the flavor, which is so common and so so likable to Indians. Actually, green tea is native, not tea, but tea as a plant is native to China. It came from China. I think this is a fact many of you would know. So here is an experiment which we did, and you go on increasing. This is the untreated cell nucleus, and then as you go on increasing, you see a comet-like structure with using 200 micromolar concentration of resveratrol. And the reason why this technique was also referred to as comet assay, because this looks like a comet. The comets, these are extra terrestrial objects which you sometimes see falling from the sky. So that's why the name comet assay. So, as I said, in the hypothesis that they mobilize endogenous copper ions. Now, we did an experiment using rats in the laboratory and generating or producing hepatocellular carcinoma, that is carcinoma of the lung in rats, by feeding them diethyl nitrosamine. And you can see here that this is normal liver, and this is the liver of a diseased animal, where you can see lesions, the cancerous lesions developing. The color of the liver is also different. And if you determine the copper levels in these cells with increasing time, you see that with increasing days of the ingestion of diethyl nitrosamine, you have increasing levels of copper in these animals. So copper, and I'll show you later some of the literature data which show that in almost every cancer, copper levels in the cells, they rise, they go up. So this is cellular DNA breakage with increasing concentration of EGCG, which is of course the most effective polyphenol found in green tea, and with increasing days, you see the level of DNA degradation increases with increasing concentrations of these polyphenols. So this is an experiment. I won't go into the details, just to show that, you see, if you use copper chelating metal chelating agents, that are specific for iron, for copper and zinc. Only copper chelating agents are able to inhibit such DNA degradation. So that shows that this DNA degradation is brought about by a combination of the polyphenol and copper ions. This is, very similar experiment which shows that with the effect of catechins on proliferation of various cancer cell lines, such as prostate cancer or pancreatic cancer, breast cancer, etc. Now here is an experiment. If you people have, I'm referring to students, I'm addressing the students. If you have heard about apoptosis or physiological cell death, you see in the body our cells are generated, they die and new cells are generated in almost every organ except probably the nervous system. So that is this phenomenon is known as the mobilization of endogenous copper which is bound to nucleus. Now I forgot to mention that overall in the cell the concentration of iron is the highest, but if you just take the nucleus, the major metal ions in the nucleus are copper and zinc. Overall is iron, but in the nucleus the major metal ions are zinc and copper. So this concentration of copper, it goes up, and this paper was cited for that particular year as the top 10 cited paper. So in conclusion, I don't want to read all of it, but the second paragraph is important. In view of the enormous complexity of the genetic origin of cancer, currently there is interest in anti-cancer research has moved to curing of cancer on the biochemistry and immunogenicity of cancer cells. In this context, our studies assume considerable significance. And if you have followed the cancer therapy research recently, immunotherapy of cancer has proved to be most promising, and there have been one or two reports that the cancer in a given individual or a patient has shown total remission, totally disappeared through immunological methods. But then this idea of ours has not been explored further because of lack of resources and the fact that I retired after some time. But it needs to be further explored. The thing is, the problem here is that plant polyphenols, which are present in vegetables, fruit, and other beverages derived from plants, as I said, they are rapidly eliminated from the body. They are, as you know, they are modified and then excreted through urine or feces very rapidly. The effective concentration of a given polyphenol is not reached in the cell to be effective. So for that, the need is to synthesize novel compounds similar to polyphenols, which would have a longer bioavailability. That means they will stay in the body, in the cell, for a longer period. So the effective concentration is reached. So that ends my thought, and thank you very much. If you have any questions,